present a method in the literature, and they say, well, this works, and here are the results, etc. Validation is quite rare. We, in a cooperation with the Biological Record Center in the UK, we carried out a validation study to test the performance of all those invented methods in the literature. We used simulation data. A number of scenarios were created, such as more visits per site over time, less field effort per site, species becoming more or less detectable over time, or bias towards the sites where the species occurs. And the question is then, how often were trends found which did not occur in the simulation data? Well, quite often. This is the type 1 errors, meaning that where you see the black arrow, this is the 5% significance level. Hopefully a proper method does not comes above this black line. And what you could see, well, here are several scenarios. What you could see is when you have a naive method, so no correction at all, you quickly have an artificial trend. Also here. And also there are some methods, even if there's no trend built in the data, it comes up with trend. So you must be careful. Well, I'm not going into the details of all these methods, but typically there are two groups. The naive methods and many of the more simple methods have too high type 1 errors. They have artificial trends. And there are some other methods, some more complex methods, which behave much better. Here's a summary of this. A simple method quickly gives bias trends in distribution, where complex methods can create unbiased trends. But there are a lot of buts here. I come back to this later. One of the methods we had covered in the simulation study is occupancy modeling, and that's my favorite sort of approach for this presence-absence data, because it's just a fascinating new sort of new class of models which has been invented over the last few years. There are some books about it. And what's the importance of this? What's the interesting of the occupancy modeling? It has two processes in the hierarchical models. One of them describing the ecological state where we're interested in, the presence or absence at a site, but also a separate submodel in it describing the observation process which accompanies the inventories always. And that's about detection and non-detection in the site. Now, I'm not entirely sure if you're all aware of the detection problem in nature, but I have an example here just to convince you that detection is often imperfect. Can I, in 10 seconds, could you raise your hands? Who's seeing the animal? 5, 4, I'm going to count to 0. But still people don't see the animal, yes? So detection is imperfect. Now let me see. It's quite an animal you have not seen here. So even in Africa, a giraffe can be overlooked. Now I give it another try. Detection, yeah, who sees it? It's not so easy to see. But it's more familiar to this sort of countries where I'm now, so you must know the species. Orca, come on. Still some people don't see it. It's not a sort of animal you should overlook, because it's a wolf. This is the habitat where you have white trees and with black spots, and you can completely overlook such an animal. Well, detection is just showing that detection is important and can vary between habitat types. I think I have time to give you a sort of a quick course in occupancy modeling, because else it's going to be abstract. So be prepared, 10 minutes or so, and you know all about occupancy modeling. Here it goes. Rabbits in the Netherlands, coastal area. 8 sites, 4 rabbits, 4 occupied sites. So occupancy is what you call the proportion of occupied sites. Here, 0.5. Half of the sites is occupied. That's clear, yes? So trends in distribution is the same as trends in occupancy, which is the trend in the proportion of sites, occupied sites. So that's clear. Yeah? That's what we're going to do. All right. In reality, let's say we have 4 occupied sites, but what we detect is not all those occupied sites. We only see 2 of them in case detection is imperfect. So we have an underestimation. So we need to take the detection into account, or else our occupancy estimate is not valid. We call this detection probability, this parameter. This is the probability to observe a species during a visit of an occupied site. So for instance, I quickly guessed the detection probability to seeing a wolf in Sweden from your observations, and that's about, I think, half or so. So during one visit, seeing a species. Yeah, there he goes. Now, a little probability theory. Let's suppose we have an occupancy probability of 0.5. So we are just putting in some rabbits, and then we have detection also 0.5, and meaning that in 2 of those occupied sites we have seen a rabbit. What's interesting that there are all sort of combinations of zeros and ones. So here there are some sites where the species is not absent and of course cannot be seen because it's not there. And you can see from the figure that you should end with 4 zeros. Present and not seen, the green ones. If you calculate it from the occupancy and detection, you could see it ends up with 2 zeros. And you have 2 ones for in case the species is present and also observed. Now what's important is that if you have occupancy and detection, then you have an expectation of zeros and ones in your observations. And if you have another occupancy and another detection probability, you will have another set of zeros and ones. That's important. Interesting question is, of course, if you only know those zeros and ones, could you from there infer your occupancy and your detection? You can, but not with 1 visit. Because if you have only 1 visit, 4 rabbits could have 8 occupied sites, or 4 rabbits could have 4 occupied sites, just depending on detection and occupancy. So that's why we talk about you need at least 2 visits. Replicated temporal replicates, that's in occupancy modeling, that's a key word, and this is the reason. You need these to separate detection from occupancy. Okay, let's try it here. Yeah, I'm not going to explain them all, but if you have 2 visits, you have occupancy and 2 detection, and you have detection per visit, you end up with all sort of detection histories generated by the visit. You have a number of red double zeros and white ones and green. You have all sort of detection histories, and these detection histories, this is what you use in occupancy modeling. Okay, 200 sites, let's give it a try. Suppose we have an occupancy 1 and detection 0.5. These are the detection histories you will expect from occupancy and detection. And if you have another occupancy, another detection, you end up with another distribution of detection histories. Yeah, I'm going too quick, or is it more or less okay? So it means when you have a different combination of detection and occupancy, you get another set of detection histories. And here's the important, you can also do it the other way around. When you know your detection histories, you can infer from there your occupancy and your detection. So this is my last slide about the occupancy quick course. Suppose you have this. This is statistics, so this is real observation. Suppose you have found in 2 visits 0, 0, and then 125, and you have from the other detection histories you have 25, 25. This matches with a detection of 0.5 and an occupancy of 0.5. But if you would have get another set of detection histories, you would get another detection, another occupancy estimation. And here comes the trick. Suppose your effort is higher in year 2 because your participants, your observers are better trained or more encouraged or whatever to do their work, then you will find more observations, but you will especially find more double ones and less of these, which means that all your observations, your increased observation effort, will go into detection rather than into occupancy. And that's the trick to take into account sampling effort for opportunistic data. You more or less, if you understand this, then you understand the trick. Okay, well, that was my quick course. Some more slides. This is about how to do it statistically. I'm not going into details now, but actually an occupancy model is a sort of logistic regression, or 2 logistic regressions coupled together. And funny thing of this is that you can have covariates to describe occupancy as in a standard logistic regression. So you say occupancy or present absence, that's what you normally say here, depends on the habitat and all sort of environmental factors, etc. But you can have separately from there, you can have detection, you can relate them to covariates. So your detection could vary with Julian date or with habitat type. You could even have a model where you have occupancy depending on habitat type and also detection depending on the habitat type. Well, that's amazing because you are not able to do it with the standard GLM, the standard regression method, to separate that sort of relations. Okay, detection histories require non-detection records. You get them for free, more or less, if you have standardized data, but for opportunistic data, you have to create them, you have to generate them. And this is the procedure how it works. Hopefully you have species list. If so, you could do this trick. Species A and B are observed, and you can infer from them that all other species have not been seen. It may not be true. They might occur there, but this is not about present absent. This is about non-detection. You simply say, well, they were not recorded there. If you have some doubts about it, you can make a more full model in your detection to do sort of fine-tuning here. Okay, well, that's all nice and interesting, of course, but the big question is, does it work, this procedure? So we validated it against the gold standard, the monitoring data, standardized monitoring data, and we ask ourselves, if we derive trends from opportunistic data for butterflies, dragonflies, and bird data, and we compare it to monitoring data, does it produce the same results? We used, well, there's some technical notes here. It's a quite big computer, unfortunately, you need to have, although in the recent years, well, this is, I think in the long run you don't need a computer cluster anymore. But these are heavy models. This is the sort of results. I of course took beautiful examples where monitoring and opportunistic data have the same trajectories. Although, on the other hand, if you look here, well, these monitoring data and opportunistic data have an enormous good correlation. So for butterflies, it worked, the trick. You have trends in monitoring data and trends in opportunistic data, and they look the same. It means that we never missed strong declines or increases, which happened in monitoring data. We could also see them in our opportunistic data. Yeah, every black spot is a species. So these are about 40 species. It also works for dragonflies. Also good correlation between monitoring data trends and opportunistic data trends. It worked for reptiles, it worked for plant species. We had a number of species groups we have processed. So we became, and we have more news, more positive news. We could also have trends for species for which we were never able to monitor using fixed standardized monitoring like this one, because this species is not, it's rare in the Netherlands, it's very mobile, you never know where it shows up. So, yeah, all positive news. So we became over-optimistic, of course, and we think, oh, okay, garbage in, trends out. That's the way we would like to see it. But here's the reality. We tried it for birds last year. We had an enormous amount of bird data. I think 5 million records or so, 10 million maybe. No, we have, I had to say, shorter time series, 8 years or so, but there was no single indication of a correlation. Whatever I tried, it didn't work, which was quite disappointing. And so here it goes, a summary like this. Yeah. Yeah. But what's interesting, of course, why does it sometimes work and why not? That's interesting. So we have an idea. We need to test it better using a new set of data. But I think this is what happens with birds. The mean species list we had for bird data were very short. We had many single records. I think 1 million single records. Here, this one, and we had well, the median was somewhere at 5 for a species list, where the average number of birds you can see in a 1 by 1 kilometer square is about, well, what is it, 30 or so, 35. So there's a big gap between these two, which means that opportunistic data, when people are in a site, they just report a few species from all the species that they could have seen. It leads to a very low detection probability, and when the detection probability is low, then these models doesn't work anymore. You get garbage. For butterflies, on the other hand, the median number on the mean number on the species list was about that, but it doesn't differ that much from the real number of butterflies. So usually when butterfly people go out, they just tick all the butterflies they see. They're not very often they're lucky because there are not so many. So just tick down, so tick a few species, and they also have the more abundant, more common ones. Detection probability is not so low for butterflies, and then it works. The same is true for dragonflies. So we believe the length of the dailies is important. Another issue is that it doesn't work for home alone species. We call them home alone because these are species that live just on their own. This is our only butterfly species living in big lots of marshland. There are no other butterflies there. So if someone reports the butterfly, then it's only then it's either it can only be this one. So it means you have no non-detection, no probability, no chance to have non-detections. The same as here for this dragonfly. This lives in the big rivers. This is not a good dragonfly area along the banks of the rivers. So dragonfly people are never going here, but only to observe dragonflies, but only for this one. So also a home alone species. So you need non-detection. So for this sort of species, it will never work. So more general, what do you need for occupancy modeling? You need temporal replicates in a period of closure. And period of closure means that a site needs to be closed for colonization or survival, but between the replicates. So these replicates need to be in the same season. You must have a procedure for non-detection records, long day lists. The longer the better. You need a number of sites for this sort of models. There's a recent paper of Magne describing if you have fewer than 60 sites, you run into trouble. Also, well, there are more model assumptions. Detection should not be too heterogeneous, and of course, if your sites are all in the southern part of Sweden and never in the northern part, your occupancy trend can never be representative for the whole of Sweden. In our case, representativity is, by the way, not so much an issue because we have so many sites that were surveyed each year. About 20% of all butterflies, of all sites in the Netherlands, we have records from each and every year. So representativity is hardly, it's almost becoming a census, so because of the density of observers. But when you have fewer observers, you need to be more careful. That's what I meant this morning. It would be helpful to give some guidance to the observers, send them to remote areas just to improve your representativity. Okay, some conclusions then. This is dangerous material, opportunistic data, but still it can be used for deriving trends and distribution if you need a sophisticated method. Be very careful with simple methods. If you have sufficient data, so try to get replicates. The replicates could be opportunistic data. Very often have many replicates because they have many observers doing the same sites in the season. Day list, that's critical also in opportunistic data. Would be good to have them. And you need to check them carefully. It will always be an issue in opportunistic data to check the results for plausibility and to see whether there's something strange happening. That's the price. That's the price of opportunistic data. But if you are successful, you get a lot of interesting results. You get distribution trends, as I described. You could also have distribution maps where you take detection into account. So this is an improved sort of species distribution modeling. You can have annual distribution maps. You can have species richness map on an annual basis. You could do much more on metapopulation dynamics using the annual maps. Here's an example of one of our butterflies. We created with occupancy modeling annual maps, and what you could see here from this species, that gradually well, blue is where the species present in 1992, and whites where they never occurred, and yellow were absent. What you could see over the years that they contracted towards more core areas. You see here, this is a blue. Here, in this is an important area for this species. In 2092, it was much more widespread here than here. So you can always see the fragmentation problems going on from year to year. Butterfly richness, it's not entirely well, but this is the sort of maps you could create for Lund. So an amazing high density of data, which you could never do with standardized monitoring data, such a spatial resolution. My last slide, yes. So far this was all about occupancy modeling, so present absence data. And I believe you can, under some conditions, you can have occupancy trends. Can you also have population trends from opportunistic data? I think as yet there's no method available to do that, where you have decent population trends. Because, well, occupancy modeling already requires quite a lot of modeling effort, but your model assumptions for dealing with abundance data from opportunistic data are much more critical. So that's going to be, well, the one who invents a method here, I'll let's say, if there's in 10 years, well, be careful what I'm saying now. Okay, I have a reward of 500 euros for someone who invents in a few years. I said, yeah, I had in mind 5,000, but then 500 euros for the one who invents a method here which really worked. So, but, no. So I think population trends really require standardized monitoring. That's what I'm saying. What is a real interesting issue for the future is combine those two types of data. I've seen several examples already where people have tried to take the standardized population monitoring data and to use the design aspects from it and use it and then to take the opportunistic data and to learn from each other in these data and to create to make a model and to do where both data were used, which could lead to much more detailed maps and trends, etc. So I think this is a really interesting issue for the future. Okay, thank you.
Thank you very much. Again, perfect timing, so we have 5 minutes for questions. Right. Yep. So, Jonathan.
Can I make an observation rather than a question that's relevant? So we talked at lunchtime about how opportunistic data such as iSpot collects have severe limitations. And I was explaining that actually we're about teaching people how to gather data. But in the process, we can produce useful data that will help you interpret detectability. So for example, we can say when people think they've seen a blackbird, 15% of the time they've seen a thrush, for example. You know, given, I mean, I don't know if that's correct, but so actually in terms
that's the false positives, you know.
Yes,
yeah,
but we can also do both and other things, and we could segment it by who's doing the we could look at variance in relation to age of observer, all kinds of things. And so actually, I think in relation to the discussion we're having at lunchtime,
I was too critical, yeah.
Instead of saying this project is no good, this citizen science project doesn't produce useful data, you should turn the whole thing around and say, actually, here's a project where you can explore the sensitivity of the data to the kinds of people and projects collecting it, and so therefore actually turn such projects into extra tools with which you can construct, the scientific the statistical correction factors.
Yeah, I like that.
And see it that way.
I agree. Yeah.
Thomas.
So detection probabilities assume has this closure assumption, and the closure assumption assume a guess that it's the same individual.
No, it's not. It's on the site level. This is detection.
It's on the site level, so it's just occupancy. It doesn't
It's
say anything
occupancy.
on
Yeah,
the-
there are two. There are two detection probabilities possible. One of them is about individuals,
Individual and
but this
another-
is just the occupancy
Okay.
on the site. So that's much more easy. So what I explained for abundance, if you're using abundance data, then one of the aspects is that your closure assumption is much more critical
Yeah, it is.
because it's about individuals.
But I mean, still it's dependent on the time period.
Yeah.
So the closure is dependent on the time period, and for birds, for example, there is a problem there because many birds, as we were talking yesterday, they stop singing, for example, and you actually detect them by song. So in your case, when you did the bird study, or there are others, how long was the time period? Because if you use opportunistic data, there is a possibility to actually use the same day, even, by the independent observer, or one day in between or so.
Yeah. Okay. Yeah. For butterflies, I just took the peak of the flight period. But for birds, it's more critical. That could be one alternative explanation why I could not find proper relation in the bird data, because the closure assumption is more critical. You're right on that. Yeah. I think I'm not quite sure, but I took the breeding season of species, and chopped off from the beginning where they start to arrive for migrants. But what I also did, I did it separately for non-migrants, because at first I expected that you can have a problem when only in a part of your site the migrants has arrived. So then it's difficult to separate detection from occupancy. But also I had no any relation with the easy to identify non-migrant species.
But it could be interesting to actually see how the detection probabilities change when you change the time window, because in the opportunistic data you have the possibility to actually do that. You can play around with the data.
That's a nice.
It could be an interesting thing to do if you read some kind of asymptote, or if it's going down quickly. Or
Yeah, that's interesting. I agree.
Dawn.
Yeah, thanks for a nice talk. I know you've also been working on estimating occupancy as a function of presence of other species that observers tend to report. Could you say something about that?
Yeah, well, I didn't explain it here, but one of the covariates in the detection model is the number of species, because that helps you to separate the quality of single records with longer lists and really long lists.
So in the bird modeling, you did account for that?
I did it, yeah.
Oh, you did? Okay.
Yeah. But the number, but I hardly had any long lists, so it was a surprise for me. There were hardly long lists in the Netherlands available. So my next step is I'm trying to get the UK data where they from about what they call the bird track data, because they say they have a lot of long day lists, and I try to repeat it and see if you have long lists, then it works also for birds.
Okay, I want to try and choose somebody who hasn't asked a question now, because all the people on the list have asked questions. So is there anybody here who hasn't asked a question and who was waving their hand around? No, actually it wasn't. So I'm now going to just randomly choose Helen to take her question, which will be the last one before our tea break.
Well, thank you very much for an excellent presentation. It's really encouraging to hear quite what you can do with the opportunistic data, and also I really like your ideas at the end in terms of coupling systematic approaches and the opportunistic approaches, because I agree agree with an earlier speaker that there's an opportunity to demonstrate to people the scientific method. Actually, many of our recorders, perhaps that's not their main motivation for being out in a live recording. So I just wondered if you had a comment as to how you might see that working, the balance of the opportunistic data versus the systematic data in terms of volunteer involvement.
You mean as compared to?
Well, I just wonder whether you think that there will be, I mean, it will vary depending on the different taxa, but whether you are optimistic that there will be next generation recording, there will be a subset, sufficient subset recording in a systematic fashion to go alongside the opportunistic data.
Yeah, well, maybe it's helpful how we treat it in the Netherlands. We have a sort of idea when we really need population monitoring, so abundance estimates, as we need for some mammals and birds, we just go for the standardized monitoring data and we forget all about the opportunistic data. But for many other species group where you could also make good use of distribution data, then it depends. So then it becomes a sort of hybrid thing. For dragonflies, for instance, if we go for both, we would like to have standardized monitoring data, but it's not so easy. So we have an addition of the opportunistic data, and the same is for butterflies. For instance, for plants, we were not successful to have a systematic design-based monitoring scheme for plants. So we now go for we go for halfway. We go for slightly opportunistic, but we steer them. We say we just don't let people collect data all over the Netherlands, but we say, oh, we do it only in these and these grid cells, and you must go there at least half a day, but the rest is for your own case. Yeah. So I think that's the way it's going to work. I believe very much in slides where possible to direct the opportunistic schemes to get more out of it. Yeah. That's more or less. Yeah.
Well, thank you very much
Yep.
again. Now we have a half-hour coffee break, so back here at 15:30.
French.